Hello everyone. My name is Dr. Lana McClements, and I will tell you today about this novel protein FKBPL that maybe quite a few of you are not familiar with. It's an anti-angiogenic protein, and my focus over the last few years has been investigating its role in preeclampsia and cardiovascular disease. So what is FKBPL? FKBPL is it belongs to the family of protein called FK506 binding proteins or immunophilins, and it stands for FK506 binding protein-like. It is a divergent member of this group, which shared homology only in the C terminal, the tetratricopeptide terminal repeat domain on the right-hand side. However, it does have homology with other members of the FKBP family, including FKBP6, FKBP52, and FKBP12, but it does not have an active enzymatic activity, which is through this domain, which is PPIA's domain, or peptidyl propyl isomerase domain. So why is FKBPL interesting? Its emerging roles over the last 10 years have shown that it is a very potent anti-angiogenic protein. Most of this work has been done in cancer, where we've shown that it can cut off the blood supply from the tumors, but it can also inhibit cancer stem cells by inducing cancer stem cell differentiation to more treatment-sensitive cells, really. And lately, we've been working and investigating its role in obesity and diabetes. It's the work that's still been ongoing, and it's been finalized at the moment. In relation to its role in the obesity and diabetes, its important mechanism of action is regulating glucocorticoid receptor signaling in the complex with HSP90, which is why it prompted us to think that it might have a role in metabolism. And in terms of its mechanism of action through CD44, which we've shown that it actually binds to and inhibits CD44 pathway, I've been investigating its role in relation to angiogenesis, but also inflammation that is emerging in cardiovascular health and preeclampsia, which is a cardiovascular complication of pregnancy. So initially, just to give you a bit of a context how the whole work in angiogenesis started. So we've shown initially that this is the work led by Professor Tracy Robson, who was my PhD supervisor and now a very close collaborator and friend. So they've shown that FKBPL, recombinant FKBPL, can actually inhibit wound closure. And also using various concentrations of FKBPL recombinant protein, they were able to show that the sprouting from the rat aortic ring was also inhibited with this protein. This was confirmed in vivo, where following sponge implantation assay stimulated by beta FGF, recombinant FKBPL was able to inhibit vascularization of the sponges. This work was published in 2011. But then we wanted to actually, what happened following this work and establishing its role in angiogenesis is that in collaboration with the local pharmaceutical industry back in Northern Ireland, Belfast, they've actually entered into a licensing deal with them, and they've developed a therapeutic peptide based on the FKBPL anti-angiogenic domain. Now, this domain was actually outside of the tetratricopeptide repeat domain, which binds to HSP90. And this is why it suggested that the mechanism is not via HSP90, but rather via CD44 protein, which was established later on. So two therapeutic peptides were designed called AD01 and ALM201, AD01 being the preclinical candidate and ALM201 being the clinical candidate, which has just completed the phase 1 clinical trials in solid tumors. What we've shown is that ALM201 is very potent at inhibiting migration and tube formation, even at much lower doses than Avastin and sunitinib, which are the well-known anti-angiogenic drugs for the treatment of cancer and other diseases affected by angiogenesis. But ARM201 does not affect proliferation, which is very interesting, but it's also able to inhibit migration of the cells when stimulated by various growth factors, unlike Avastin and sunitinib, which can only inhibit migration stimulated by VEGF, and sunitinib also PDGF. In tumor models, AD01 was able to inhibit vasculature around the tumor and that way inhibit tumor growth. So following from this work, what we wanted to find out is that since FKBPL is such a potent anti-angiogenic protein, what are its physiological roles? So this work was funded by BBSOC in the UK, and we established a model of FKBPL knockdown mice to investigate its role in physiological and developmental angiogenesis. To our surprise, full knockout of FKBPL was embryonically lethal, suggesting its critical role in developmental angiogenesis, which has led us to the work in preeclampsia, which is associated with developmental angiogenesis. But once we've established the heterozygous knockdown of FKBPL, the embryos were viable, but they did show early signs of endothelial dysfunction and vascular integrity, as demonstrated here in the red by endomucin staining. So there was a vascular disorder and poor integrity then in the wild type mice. And further from there, we've characterized the model in greater depth and show that, yes, indeed, FKBPL levels were reduced in all organs in FKBPL mice. And whilst the phenotype was pro-angiogenic, which was expected given that FKBPL is anti-angiogenic protein, so when we knock it down, it becomes pro-angiogenic with increased number of vessels, for example. We were able to actually ameliorate and restore the phenotype by treating these mice with AD01 peptide, which is a therapeutic peptide mimetic essentially of FKBPL. So that was encouraging, that we could actually restore the low levels of FKBPL and the phenotype in those circumstances with our drug. So in terms of cardiac function in FKBPL mice, we proceeded to investigate this, and this is the work completed by my previous PhD student, Dr. Abdulrahim Al-Qudar. We were looking at 22-week-old mice in terms of the cardiac function and cardiac phenotype. Inflammatory markers were actually increased in the hearts of FKBPL mice, which is demonstrated here by the stronger green color. A number of these factors were actually increased, so it showed a pro-inflammatory phenotype, cardiac phenotype, and in association with this, there was the presence of cardiac hypertrophy as well. Now, this cardiac hypertrophy could be protective, but we need to explore this further. What we are also interested in, that whilst this initial inflammation or pro-inflammatory phenotype in the heart could be protective, longer term and persistent inflammation could actually lead to further cardiac damage and eventually heart failure, which is what we are actually investigating at the moment. Further to this, we've also shown that in FKBPL mice, in the heart tissue, there was increase in ICAM1 expression, which is a marker of endothelial dysfunction, suggesting to us that actually, whilst there is a myocardial hypertrophy, there are signs of poor vascular integrity and potential endothelial dysfunction. So in summary, this first part of the talk, I've shown you that FKBPL is a novel anti-angiogenic protein, and its therapeutic peptides have been designed and are currently in the clinical trials. But also FKBPL has a critical role in physiological and developmental angiogenesis, which is what's leading us to the next part of the talk, and its role in preeclampsia. However, low baseline levels, whilst pro-angiogenic, they show compromised vasculature and signs of endothelial dysfunction, as well as cardiac inflammation and hypertrophy. So following from this work, we've wanted to investigate the role of FKBPL in preeclampsia. So before I get into all of this, what is preeclampsia for those of you who are not too familiar? Preeclampsia is a very dangerous condition in the latter part of pregnancy. It is characterized by high blood pressure, the new onset of high blood pressure, post 20 weeks of the gestation, and organ damage often involving liver or kidneys. It occurs in about 5 to 6% of pregnancies, and it is on the rise, unfortunately, because of the increase in aging mothers, also obesity and diabetes, which are the risk factors for this condition. But not only that, preeclampsia also has long-term complications, and women with preeclampsia end up, 2 out of 3 women with preeclampsia end up dying of cardiovascular disease later on in life. What is even more concerning is that there are no effective treatments or monitoring strategies are lacking really for this condition, particularly for the late onset preeclampsia. Now, there are 2 types of preeclampsia currently defined. So preeclampsia is a multifactorial and a complex disease, and we're still trying to understand its pathogenesis. But what we know so far is that the early onset preeclampsia comprises about 10 to 15% of cases, of all preeclampsia cases. It occurs before 34 weeks of gestation. It is characterized by the growth restriction of the fetus, whereas late onset preeclampsia is more sort of related to the underlying maternal issues such as obesity, vascular dysfunction, etc., and does not necessarily lead to smaller babies, for example. But to put everything into perspective, preeclampsia still kills over 70,000 women and 500,000 babies every year. And most of these deaths occur in developing countries due to the poor prenatal care, which is critical in picking up this condition early and preventing complications. Complications in pregnancy include preterm birth, HELLP syndrome, which is fatal, eclampsia, stroke, and a number of these complications actually can be fatal if not managed properly. But also in terms of the fetal distress, there is a risk of neonatal respiratory distress syndrome as well. And long-term complications include diabetes, cardiovascular disease, and stroke. So it's not only important that we manage these women well during pregnancy, but also after pregnancy for the long-term cardiovascular effect. So what do we know about preeclampsia? Again, it's a very complex disease, and we're trying to understand the molecular regulation of many processes. But what we do know is that initially, at the very start of pregnancy, these cells, fetal cells called trophoblasts, they invade spinal uterine arteries of the mother, which are sort of in normal pregnancy, like sort of dilated, elastic, and allow unlimited supply of oxygen and nutrients to the baby through the placenta. However, in preeclampsia, the remodeling is actually restricted. And this leads to restricted supply of oxygen and nutrients to the baby, and that can lead to growth restriction. But it can also lead to placental dysfunction, and which further on can affect the systemic vascular function as well. And as a result of this, hypertension can occur towards the end of pregnancy. But what is very important to understand at this stage is that preeclampsia actually starts to develop early in pregnancy, but we're only able to pick it up much later when it's a lot more dangerous. So from what we know about preeclampsia is that angiogenesis plays a key role, abnormal trophoblast invasion. There is hypoxia in the placenta because of this limited remodeling or poor remodeling of the spinal uterine artery and a restricted supply of blood and oxygen and nutrients, and there is altered immune response. In fact, FKBPL plays an important role in all these processes, which is why we endeavor to investigate its role in this process. So in conjunction with its target protein, CD44, we actually wanted to measure the levels of these 2 proteins in plasma of the patients or women who proceeded to develop preeclampsia, but they didn't have any symptoms of preeclampsia, which means that we measured the levels at the 15 and 20 weeks of gestation, which is pre-diagnosis of preeclampsia, and then these women were followed, and the ones in red developed preeclampsia, and the ones in black didn't develop develop preeclampsia. Now, this was done in collaboration with Professor Louise Kenny from University of Cork, and these samples are part of the famous SCOPE study. So when we measured the levels of CD44 and FKBPL, the ratio was actually increased in those women that proceeded to develop preeclampsia. Then in our collaborators from the Mayo Clinic, we wanted to check what actually happens once women are diagnosed with this clinical condition. So this is post-diagnosis towards the end of pregnancy, before women are giving birth. And in fact, opposite was seen, which suggested to us that there might be some longitudinal changes before preeclampsia is actually diagnosed and fully developed, to when actually is fully clinically developed. So this suggests to us that low CD44 FKBPL ratio suggests a restricted angiogenesis, whereas high CD44 FKBPL ratio suggests pro-angiogenic phenotype, which could be compensatory at this stage in pregnancy. And this is exactly what we are thinking, that hypoxia as a pro-angiogenic stimuli would increase CD44 levels, which is pro-angiogenic protein, and reduce FKBPL levels, which is anti-angiogenic protein. But in doing so, as I have shown you in the first part of the talk, it could be causing endothelial damage, which is one of the well-known causes of preeclampsia. And then once preeclampsia kicks in, the levels of FKBPL increase and the levels of CD44 decrease, and that is an anti-angiogenic state. Now, we have interest in how to correct this balance, and one of our research interests are in developing mesenchymal stem cell treatment as a potential novel treatment for preeclampsia that I will talk about in a few slides. Also, we've shown that FKBPL has a role in placental development. So these are women with preeclampsia, PE, and without preeclampsia. So sort of aligned to what we've seen in the plasma of women who developed preeclampsia or who had established preeclampsia, also levels of FKBPL were increased in placenta, indicating anti-angiogenic placenta or ischemic almost placenta. And this was confirmed at the microRNA level. So FKBPL levels were increased and CD44 decreased, and in association with signs of endothelial dysfunction. So then, in terms of the endothelial dysfunction, what we wanted to see is how does hypoxia regulate angiogenesis, which is well known, but what can FKBPL do to restore the angiogenesis? So outside of hypoxia, FKBPL does inhibit angiogenesis, which is a well-known fact. But what is interesting is that actually in hypoxia, overexpression of FKBPL is able to restore angiogenesis to the normal level and bring that pro-angiogenic effect down, which is very interesting and very important finding. In terms of the trophoblast function, FKBPL overexpression is actually able to stimulate that clonogenicity of trophoblasts, which is important for placental development. And this is the work we are continuing to investigate, and we are developing for this purpose first trimester trophoblast organoids, which are the 3D model of the placenta, in which we are actually hoping to see the effects of FKBPL, and we are establishing a clone of this first trimester trophoblasts where we've knocked down FKBPL using CRISPR technology, and we're almost there with the full knockdown, which we're taking forward in characterizing its role in placental development. So just briefly before I finish my talk about the mesenchymal stem cells, so this is the review that we've published recently, current hypertension reports suggesting the mesenchymal stem cells could be the new potential treatment for preeclampsia due to their role in improving angiogenesis, in inhibiting inflammation, and also oxidative stress, which are all important processes in preeclampsia. But not only that, mesenchymal stem cells, the condition media for mesenchymal stem cells is likely responsible for this pro-angiogenic effect, and most likely extracellular vesicles within this condition media are the ones that are actually stimulating and increasing and improving angiogenesis. And concurrently, by improving angiogenesis, they're actually able to affect the expression of FKBPL and CD44, which is why this is particularly interesting to us. And it appears that actually mesenchymal stem cells are able to reduce the expression of FKBPL at a protein level, mostly in normoxia, not so much in hypoxia, and concurrently increased levels of CD44. So this is also very interesting because this hasn't been shown before in terms of FKBPL signaling. So in summary, I hope I've convinced you that FKBPL and its target protein CD44 have diagnostic potential in preeclampsia, both for prediction but also diagnosis. FKBPL appears to regulate trophoblast and endothelial cell function, and its levels are critical to the normal cellular functioning, and potentially have implications in the pathogenesis of preeclampsia. And mesenchymal stem cells, via their secretome, could be new treatment for preeclampsia by reducing the levels of FKBPL and increasing CD44, which are part of the pro-angiogenic mechanism. So this work has been done in collaboration with many wonderful collaborators I have here in Australia, but also internationally, and also my lab members at QUB, Queen's University Belfast, and UTS. So thank you for listening, and I'd like to welcome any questions.
Thank you very much, Lana. It's a really interesting area and great presentation. So we have a few questions here. So the first one from Sandra is, did you differentiate between pregnancy-induced hypertension, PIH, and preeclampsia?
Very good question. No, we haven't done that yet. So it's certainly something that could be interesting. We have some data to show that angiotensin 2, for example, which is obviously elevated in hypertension, regulates expression of FKBPL, and that ADO1 can actually, which is FKBPL peptide, can abrogate cardiac hypertrophy induced by angiotensin 2. So certainly interesting area, and potentially with application for FKBPL.
Beautiful. I'll follow up with another question, I guess. So it's really interesting to see your organoid working really well in this area. How is there a good preclinical animal model that's valuable for preeclampsia?
That's a great question, Helena. Thanks very much for that. Actually, preeclampsia research has been hindered by the lack of reliable models. So this is an area that we are interested in. We're trying to kind of develop patient-derived models or first trimester placenta 3D models that can be used, because animal models actually, animals don't develop preeclampsia spontaneously. It has to be induced. And hence, there are a lot of features that are not really translatable to human disease. Again, there are many different models as well. We are using the RAP model, which is the reduced uterine perfusion pressure model. We haven't had time to actually present data on, but I do have some data on that one. So that's probably the most reliable one at the moment. But again, not very close to human disease.
Right. Thank you. I got another one. Is FKBPL also affect lymphoangiogenesis, or is angiogenesis specifically?
We don't know about that. We've looked at the angiogenesis in particular. So pathological angiogenesis mainly in the context of cancer. And then now we are looking into the pathological angiogenesis in cardiovascular disease as well. So I'm not entirely sure about lymphoangiogenesis yet.
Great. Thank you. I think there's another one. So fascinating talk. Given the differences you see in FKBPL levels and the different stage of pregnancy, but was wondering if you have considered generating an inducible FKBPL mouse model so you can manipulate the levels at specific times during pregnancy.
That would be great. If you want to get in touch, I'll be more than happy to discuss this option. Certainly is a very good concept and very important, because the levels seem to, high levels or low levels seem to both be bad. So I think it's very critical that the levels are kept at the right level, at the right physiological level, because bringing them up or down, it seems to kind of cause trouble either way.
Great. Thank you. We got another question from what kind of MCS are the best candidate for preeclampsia?
That's a great question. So in our review, we've shown that in preclinical models, there are about 5 or 6 studies. They've shown that mesenchymal stem cells from bone marrow, but also from the umbilical cord, can actually ameliorate symptoms and features of preeclampsia. We are focusing on bone marrow at the moment, but also we have some potential candidates from the residual placental mesenchymal stem cells and adipose tissue stem cells that could be good candidates too.